Okay, it's 8 o'clock. Let's get started. Greetings and welcome to all of you to the ZOOMinar. Today we have two speakers, Dr. Masaki Okumura from Tohoku University, and the second speaker will be Dr. Takahiro Murakoa from Tokyo University of Agriculture and Technology from Japan. Thanks for joining us. I know the timing is not that good for you guys, but I really appreciate it. The first speaker is Dr. Okumura. He has completed his PhD degree from Kwansei Gakuin University in Japan in 2011. He worked on oxidative protein folding under the guidance of Dr. Hiroshi Yamaguchi. He was a JSP's postdoctoral fellow in Kyushu University. And then in 2016, he joined Tohoku University as an assistant professor and was promoted to the rank of associate professor recently in 2022. His research interests include protein folding, protein aggregation, and amyloids using structural and biochemical approaches. So with that introduction, I would like to welcome Dr. Okumura-san.
Okay, I'm Masaki Okumura from Tohoku University. So I would like to thank Professor Rams for giving me opportunity to talk my recent study, and thanks for kind introduction. So today's my talk is how disulfide bonds are introduced into protein. Disulfide bonds play an essential role in cell viability. Protein disulfide bond is covalent bond. Covalent bond formed between two cysteine residues. As indicated in here, BPTI, lysozyme, and insulin have a different number of disulfide bonds. And these disulfide pairings are essential for maintaining native protein conformation and bioactivities. Although it has been well studied, the in vivo pathway of disulfide bond formation and folding is still unknown, even in insulin. So, Anhuizen performed refolding experiment of reduced denatured RNA, implying that protein folds spontaneously under thermodynamic control. Then he was winner of Nobel Prize in chemistry, as you well know. However, it takes several hours for refolding time. Actually, when adding a microsome, disulfide bond formation was accelerated dramatically. So microsome contained endoplasmic reticulum, ER, one of organella. When over 30% of total protein is inserted into ER, disulfide bond formation mainly occurs. It was found that protein disulfide isomerase, PDI, introduces a disulfide bond into substrate. Therefore, ER controls protein homeostasis. So ER is an essential organelle where secretory and membrane protein are newly synthesized and falls into native conformation containing native disulfide pairings. It is well known that PDI constitutes a primary disulfide bond formation pathway in the ER. While PDI play a central role in oxidative folding in the ER, recent studies suggested that other pathways by 20-member PDI family also exist in mammalian cells. Therefore, the oxidative folding network is even more complicated than previously thought. There is another interesting aspect related to misfolding pathology. Several studies reported that PDI family members closely related to various neurodegenerative disorders. This summarizes the relationship between PDI family and misfolding disease in this table. In this report, PDI family might associate to progress of pathogenesis and thus mutations of PDI family leads the dysregulation of their function regarding the protein homeostasis. So PDI family members commonly contain at least one thyroidexin domain with redox active CXXC motif, but vary in number and three-dimensional arrangement of thyroidexin domain. Presumably, these different structural features lead to different function of PDI family members. However, it still remained unclear how PDI family recognize a wide variety of substrate with different shape, size, and number of disulfide bonds. To reveal substrate recognition mechanism by PDI family, it seems to be better strategies to employ several approaches, such as a single molecule, a solution, a crystallographic, and cryo-EM, I think. Recently, we demonstrated a new structural information of PDI at the single molecule level. Single molecule approach by high-speed FM demonstrated that only oxidized PDI repeat cycle between monomer and dimer in the presence of unfolded clients. First topic is PDI catalyzed folding mechanism. Herein, I explain PDI function along client folding. At the first step, oxidized PDI act on unfolded client to introduce disulfide bonds. Thereafter, reduced PDI traps folding intermediate and catalyze disulfide isomerization. PDI can catalyze oxidative folding of various clients like lysozyme, PPTI, and RNSA. However, the mechanism of how PDI guide proper oxidative protein folding of various clients remains unclear. Based on crystal structure of human PDI, reduced PDI forms a closed U shape, while oxidized PDI forms a more opened U shape, providing a more expanded pocket. Probably, various clients are captured by this central pocket. However, nobody has directly observed the PDI action during the catalysis of oxidative protein folding. To confirm redox-dependent conformational change of PDI in solution, we first carried out SAX analysis for reduced and oxidized PDI. RG and Dmax were determined by SAX measurement, showing that oxidized PDI assume a more opened conformation compared with reduced one. However, the measured scattering profile did not overlap with those calculated from crystal structure. This result suggested that there is significant difference in domain arrangement between solution structure and crystal structure. So to visualize the reduced and oxidized PDI at a single molecule level, we next employed the high-speed AFM. This method was developed by Professor Ando from Kanazawa University. Compared with conventional AFM, Professor Ando succeeded in high-speed scanning. Therefore, it is possible to observe the movement of molecules by time resolution of 0.001 sec and nanometer order resolution. This slide shows you AFM image of reduced PDI when measured long and short axis and the height of several hundred molecules. Reduced PDI showed a single population with a long axis of 86 angstrom, which should correspond to close conformation. However, in the case of oxidized PDI, a long axis showed two populations of 82 angstrom and 110 angstrom. These results indicated that oxidized PDI has at least two conformations, open form and closed form. Here is shown a time course of long axis of same PDI molecule every 0.1 sec per frame. We found that reduced PDI holds a closed conformation persistently, while oxidized PDI showed interconvertible, opened, and closed conformation. Thus, high-speed AFM analysis demonstrated at the single molecule level that PDI actually undergoes striking redox-dependent change in thyrodoxine domain arrangement. In order to elucidate the client recognition by oxidized PDI at initial folding step, I prepared all cysteine blocked BPTI modified by maleimide. BPTI 3-disulfide bond contributed to its conformational stability. Thus, this all system blocked BPTI cannot fold and maintain random core structure. Interestingly, upon addition of system blocked BPTI, PDI converted to a dimer by assembling face to face to create a central cavity, as shown here. And dimerization depends on concentration of system blocked BPTI as the unfolded model substrate. This is a reduced trapping mutant of PDI. This mutant does not dimerize even at a high concentration of system blocked BPTI. Therefore, just only oxidized PDI assembled to a dimer in the presence of system blocked BPTI. In agreement with this finding, when adding system blocked BPTI to oxidized PDI forms to a dimer in the bulk experiment as observed by SEC-Mars. Consistent with the result of single molecule, unfolded BPTI induces PDI dimerization in solution in a dose-dependent manner. Okay, next, we added reduced BPTI as a normally folding substrate to oxidized PDI and measured AFM image of PDI. You can see when mixing reduced denatured BPTI, PDI showed repeated cycle of dimerization and dissociation to monomer. In the presence of unfolded substrate, unfolded BPTI, oxidized PDI repeat monomer and dimer at the interval of about 0.5 sec. We next prepared a cysteine blocked folding intermediate. Briefly, folding intermediate N star is purified by reverse phase HPLC, and next free 2-cysteine residues of N star are modified by maleimide. Therefore, cysteine blocked N star cannot convert into native conformation. Of course, cysteine blocked N star is stable folding intermediate. This slide shows you oxidized or reduced PDI dimerase depending on folding status of substrate. Reduced PDI hardly forms a dimer in any folding state of client. However, oxidized PDI forms a dimer when substrate is in more collapse conformation. Thus, just only oxidized PDI can detect folding status of client and forms a dimer. As a generality of client recognition, oxidized PDI also forms a dimer in the presence of unfolded RNSA, another substrate bigger than BPTI. Probably its substrate capacity is restricted due to the limit of cavity size. Therefore, our next interest is how PDI recognize much larger substrate than RNSA. To explore this, we employed plasminogen with 7-ly disulfide bond as a bigger substrate and visualize action of PDI on unfolded plasminogen by high-speed AFM. Reduced and denatured plasminogen was reacted with maleimide and cysteine blocked plasminogen cannot fold. AFM image, each image of cysteine blocked plasminogen indicate its extended and flexible conformation as shown here. Of note, when we added oxidized PDI to plasminogen, two PDI dimers bound an unstructured chain of plasminogen. I speculate that two dimers of PDI may act on unfolded client and catalyze the oxidative folding of local conformation. Therefore, substrate-induced PDI dimerization is a general event observed with reduced and denatured client other than BPTI. So this is a first section summary. This study reveals that PDI assembles to a dimer during the catalysis of oxidative protein folding. We believe a PDI capture unfolded client in this cavity and catalyze its oxidative folding efficiently. Thus, we propose a new mechanism of operation of PDI, which was first clarified by single molecule observation using high-speed AFM. Okay, second topic is new structure and function of P5, one of PDI family. Recently, we demonstrated that P5 rapidly introduced a disulfide bond into substrate during initial folding step. We determined the structure of full-length P5 by Sachs experiment and crystallographic study. This molecular modeling reveals that P5 dimerize in solution via unique leucine zipper motif. P5 consists of 3 thyrodoxine domains. First and second domain contain redox active CXXC motif, and I prepared A0A or AB truncated mutant. Interestingly, SEC analysis indicated that elution volume are 12 milliliter for A08 truncated mutant and 15 milliliter for AB truncated mutant, suggesting an oligomeric state of A0A via A0 contact. Next, we carried out SAX analysis for reduced and oxidized P5. Both measured scattering profile and PR function of oxidized P5 overlapped with reduced one. This result suggested that there is no significant difference in domain arrangement between reduced and oxidized P5. The molecular mass calculation using BSA as a standard is about 100 kilodalton, indicating a dimer in solution. Furthermore, estimated molecular mass are 70 kilodalton for A08 truncated mutant and 40 kilodalton for AB truncated mutant. Based on these evidences, P5 forms a dimer via A0 domain. The radius of gyration of P5 were also estimated to about 47 angstrom. This parameter is larger than ERP46 and PDI. Therefore, P5 assumes to exist in an extended manner. We next try to refine ensemble optimized model of P5 using crystallographic and SAX data. Several models of P5 were solved by single rigid body model. These data indicated that P5 forms a dimer via A0 domain, and 3 thyrodoxine domain adapt a flexible conformation in an independent manner. When we focus on A0 crystal structure in detail, we found out outside amino acid radius of alpha helix 4 in A0 domain is leucine-rich region. When we confirmed amino acid sequences in several species, we found it to be highly conserved, here indicated as yellow. Thus, I tried to create 5-ARA mutant for the preparation of monomeric P5. Elution volume of 5-ARA mutant by sex shifted to 15 milliliter. Briefly, SAX analysis clearly indicated that this P5A5 mutant is a monomer in solution. Therefore, P5A0 domain contains a leucine zipper-like motif. It remains unclear dimerization function of P5. First, I tried to estimate oxidative folding ability. This is traditional oxidative folding assay. BPTI is a model substrate because folding pathway is determined in detail. This assay enables us to determine disulfide bond position with structural information. For example, 2 disulfide bonded N' and star NSH species are partially folded. Therefore, this assay is easy and useful for chasing folding reaction. Regarding P5 catalyzed oxidative folding, I employed BPTI as model substrate. The result indicates that P5 and P5A5 rapidly introduce disulfide bonds into BPTI than TDI during early folding step. Therefore, monomeric P5 has the ability to introduce disulfide bond into BPTI at almost the same rate as wild type dimer. Next, we try to estimate P5 regulated IRE1 oligomer. I explain briefly IRE1 regulated unfolded protein response UPR cycle. Regarding regulating IRE1, in non-ER stress, IRE1 exists a non-covalent dimer as an inactivated state. When misfolded protein accumulate, IRE1 forms a higher oligomer. The oligomer is stabilized by not only hydrophobic interaction, but also intermolecular disulfide bonded. Activated IRE1 promotes the expression level of messenger RNA chaperone proteins such as PDI and BIP. And when decreasing ER stress, P5 reduces intermolecular disulfide bond in the IRE1 oligomer in this inactivation step. This slide shows you time course of IRE1 inactivation in vitro. IRE1 luminal domain was obtained by E. coli expression system and purified by monoQ and sec. In the presence of DTT, oligomeric IRE1 was dissociated into non-covalent dimer of IRE1. Interestingly, dissociation into non-covalent dimer IRE1 was accelerated by wild type P5. However, monomeric P5 mutant slowed down this reaction. Therefore, dimerization of P5 appeared to control IRE1 inactivation step. Finally, we confirmed conformation stability of monomeric mutant. PyUVCD indicate content of protein secondary structure such as alpha helix and beta sheet. Compared to the wild type, alpha helix content of monomer P5 is reduced, suggesting a partial unfolding due to the collapse of secondary structure. NMR measurement were performed and each radius was assigned with and without leucine zipper like adhesive motif of first domain involved in dimer formation. This shows a chemical shift, and when chemical shift is mapped to its structure, not only chemical shift of alpha helix 4 related to dimer, but also other local conformations such as alpha helix 2 undergoing chemical perturbation. This result shows leucine zipper-like adhesive motif is involved in overall structural stability. Monomeric P5 may cause aberrant protein due to conformational destabilization in the cell. Therefore, we test the effect of P5A5 overexpression in HeLa cell. Then it was found overexpressed P5A5 induces the increased expression level of BIP as compared to wild type P5. This is probably because P5A5 was treated as an aberrant protein in the ER due to conformational destabilization. So this is a summary in this work. P5 forms a dimer via A0 domain, and 3 reduction domain adapt a flexible conformation in an independent manner. Dimeric P5 efficiently promotes the reduction reaction of IRE1 oligomer, leading to inactivation. Monomeric P5 has a lower ability to cleave the disulfide bond in IRE1 oligomer. Monomerization induces artifact intermolecular disulfide bond into dimer and structural destabilization. Therefore, P5A5, a monomeric mutant, is treated as an aberrant protein in the ER due to conventional destabilization, induces IER stress, and increase expression level of chaperone BIP. So I would like to acknowledge Professor Inaba for my mentor. And also, I greatly thank my group, Dr. Kanemura, Dr. Matsuzaki, Dr. Kinoshita, and Miss Fukamachi, Miss Sato, and Miss Watabe for great support. This work was also performed in collaboration with Professor Ogura and Dr. Noi for high-speed FM, and Professor Akiyama for SACS. This work was also performed in collaboration with Professor Sayo for NMR and Professor Lee for CD or ITC. Although today I did not show the data in today's presentation, I would like to thank my international mentor, Professor Burate and Professor Murakoa for chemical regulated protein, oxidative protein folding. So I'm sorry, time was past. Thank you for your attention, and I would be happy to field any question or comment.
Thank you very much, Okumura-san, for a very nice talk. Real nice results. Let's wait for maybe 30 seconds before we see the questions in Q&A. Let me ask a question to get started. In your P5, this oligomerization driven by intermolecular disulfide bond. How big are the oligomers? Can it form higher order oligomers?
So in wild type, P5 forms a dimer, non-covalent dimer, non-covalently forms a dimer via A0 domain. So via leucine zipper-like motif. So, but P5A5 monomeric mutant forms a oligomer, disulfide bonded oligomer. So this disulfide bond is artifact.
I see.
Artifact. Yes, artifact. Artifact disulfide bond.
But does it lead to the formation of higher order oligomers? Maybe sometimes amyloid fibers or anything like that?
Sorry, I don't check.
Okay. What about the metals? Do they have any role to play, like zinc, copper, because they can bind to cysteine?
Yes, it is very good question. So, so in the ER, major ion is calcium ion, and in the secretory pathway, we know secret pathway, zinc ion is most important. So especially in Golgi. So in the ER, there are no ion such as ferrate or maybe copper. So in the ER, it is most important about calcium ion, I think.
Okay. Okay, I see a question in the Q&A from Velayudam. What about the interaction between proteins containing disulfide bond and H2S? Do they form polysulfide bond?
So I cannot detect polysulfide. So it is a very good question, and it is very good problem. Yes, I think.
Danilo, you have a question?
Yeah, just very quick questions about BPTI, because BPTI is a quite interesting protein because it is one of the rare examples of protein that unfold according to a downhill, in other words, a barrierless folding mechanism. So you elegantly depicted how the disulfide bridges may have a role in driving this kind of folding mechanism. So my question is, is it possible to isolate BPTI intermediates with different amounts of oxides reduced and study the fold instability of these intermediates in an attempt to clear up how they can help to drive the downhill folding mechanism, which is a quite mysterious folding mechanism.
Yes, yes, I see. I think so. So now I can separate folding intermediate or VPTI or any other client such as insulin. So, but it is very difficult to understand about protein folding intermediate. So, okay. So that's good point, I think.
Okay, thank you.
Thank you.
Okay, there are no other questions I see in Q&A. Thank you very much, Dr. Okumura-san, for a very nice talk. Let's shift to next speaker, Dr. Murakoa. Can you share your screen? Okay, Dr. Murakoa received a bachelor's degree in engineering in 2002 and a PhD degree in 2007 from the Chemistry and Biotechnology Department at the University of Tokyo. He was a JSPS postdoctoral fellow in Professor Samuel Stoops' laboratory at Northwestern University, USA. He then returned back to Japan to join Tohoku University as an assistant professor in 2008. He was promoted to associate professor rank at Tohoku in 2017. Since 2020, he is a professor at Tokyo University of Agriculture and Technology. He has received many awards, including the Innovo Research Award in 2009, Chemical Society of Japan Award for Young Chemist in 2015, the Young Scientist Award in 2016, the ESPEC Foundation Award in 2018, and the Mazda Foundation Research Award in 2019. So with that brief introduction, I would like to welcome Dr. Murakoa-san to give you a talk.
Thank you very much for the kind introduction, Professor Ramos. And it's my great pleasure to share my recent study in this ZOOMinar. So I'd like to talk about the development of functional material, particularly the supramolecular material for the injured brain regeneration. Yep. So the extracellular matrix is a tissue that surrounds cells by forming a networking structure made of fibrillar proteins, mainly the collagens. Actually, the ECM is one of the central tissues that regulates the cellular activities. And for that activities, the ECM plays three major roles, as I show here. So the first is cell adhesion activity, which allows for regulating the cell migration. And the second function is scaffolding, which is a mechanical function of the ECM. And the third is signaling, which regulates the cellular activities such as regeneration, differentiation, and proliferation. And the signaling function is performed by the sustained release of the growth factors from the ECM. So inspired by such attractive functions of ECM, many researchers so far have developed the synthetic mimics of ECM for tissue engineering. Among numerous ECM mimetic biomaterials, self-assembling peptides that form supramolecular nanofibers are promising because of their biocompatibility and biodegradability. Here are the representative and pioneering examples. Sorry. of the ECM mimetic peptides reported by Chen and Stupp. Both peptides are amphiphilic to form supramolecular nanofibers in aqueous media and show cell adhesion and scaffolding functions analogous to the ECM. Although these materials are effective for cell culturing due to these two functions, there have been no reports of fiber-forming peptides that show signaling functions by sustained release of growth factors using full-length proteins. Coulier and co-workers have reported the incorporation of full-length proteins into the supramolecular peptide nanofibers. However, they report that the incorporated proteins are hardly released from the fibers, likely due to the highly sticky and aggregative properties of these self-assembling peptides. Even after the 120 hours, the proteins are still remained within the peptides. Recently, our group has developed the first example of the fiber-forming peptides that show cell adhesion, scaffolding, as well as incorporation and releasing functions of full-length proteins. And because our material possesses all these functions that ECM performs, it is very functional for tissue engineering, and we could successfully demonstrate injured brain regeneration in vivo. So extracellular matrix consists of network structure of the fibrillar proteins, and a similar structure can be constructed by self-assembling peptides capable of one-dimensional stacking. And for promotion of tissue regeneration, sustained release of growth factors is necessary through the time scale of several hours or even days that matches the period of regeneration. As the fiber-forming peptides, the supramolecular peptides, RADA16 or RADA16 is a commercially available aphilic peptide, and it is maybe one of the most popular materials, even including the clinical applications. The RADA16 consists of repeating amino acid sequence between the hydrophilic amino acid residue and hydrophobic alanine residue, which affords the hydrophilic phase as well as the hydrophobic phase, allowing for self-assembling and beta sheet formation, providing the nanofiber. Although the RADA16 forms quite stable hydrogel, as I show you here, it has several limitations, such as formation of non-specific aggregation and heterogeneous property. Here is a confocal laser scanning micrograph of fluorescent labeled RADA16 showing micrometer scale aggregation. And this is most likely due to the slipping of the hydrophobic interaction here between the alanine phase because of the flat surface of the hydrophobic phase. The perfectly matched face-to-face stacking allows for the homogeneous fibrillar structure. However, such a slipped assembly results in the formation of non-directed, the multi-dimensional aggregates, non-specific aggregation. For the directed, the regulated assembly, we designed a novel peptide called ZIGZAP. So ZIGZAP forms a highly transparent hydrogel with homogeneously dispersed property in a micrometer scale or even larger scale in a physiological condition. And such ideal gelation properties are realized by slow and site-specific assembly by the conformational change of the peptide, as well as the dovetail type stacking between the hydrophobic layers of the peptide, which is due to the characteristic jigsaw-shaped hydrophobic domain made of the bulky isoleucine and methionine residues and smaller alanine residues between them. The molecular structure of ZIGZAP was inspired by a characteristic structure found in membrane-spanning proteins. Membrane-spanning proteins with forming a helix dimer structure, such as glycophorine A, are known to have characteristic AXXA or GXXA motifs. And these motifs can transform the secondary structure from helix to the beta sheet in response to external stimuli such as heating to provide the dovetail packing. By mimicking this motif with a modification to include an amphiphilic property to the peptide, we developed finally this molecule as ZIGZAP that contains AXXA motif, as we can see in the natural membrane-spanning proteins. So as seen in the protein, the membrane protein, the ZIGZAP also showed a conformational change from the helical to the beta sheet structure as visualized by circular dichroism spectroscopic measurement. And during the transition, ZIGZAP increased the elasticity up to 10 to the minus 4th Pascal and formed micrometer long nanofibers as visualized in TEM. So with a fiber-forming peptide having a dynamic property in hand, we tried to develop a supramolecular material that can incorporate and release the full-length protein, particularly VEGF, vesicular endothelial cell growth factor, as a mimic of ECM. And for facile characterization, we synthesized ZIGZAP tagged GFP as well. The ZIGZAP tagged GFP was efficiently incorporated in the ZIGZAP hydrogel, while the non-tagged, just simple GFP was hardly incorporated. The incorporation efficiency was quite low without the peptide tagging. Analogously, the incorporation efficiency of the ZIGZAP tagged VEGF here was significantly higher than that of non-tagged the naked VEGF. And therefore, it is demonstrated that the GIGSAP tagging, the peptide tagging, is very useful for the supramolecular incorporation of the full-length proteins. And importantly, the incorporated GFP with the GIGSAP tagging showed the green fluorescence, suggesting the formation of the native structure in the hydrogel. The incorporated GIGSAP tagged GFP and also the GIGSAP tagged VEGF, was successfully released from the hydrogel over the 100 hours. It's good for the biomedical applications. And in contrast, the RADA16 tagged protein was hardly released from the hydrogel, and this result is consistent with the previous report, which shows the remaining of the GFP within the hydrogel over 100 hours. So therefore, in vitro conditions, we demonstrated that the full-length proteins with GIGSAP tagging can be efficiently incorporated in the hydrogel, and the incorporated proteins also shows sustained release from the hydrogels over the 100 hours. And the GFP also formed the hydrogel in physiological conditions such as in serum. Yeah, and in addition, in a tube forming assay using HUVEC, the GIGSAP hydrogel containing the GIGSAP tagged VEGF showed the formation of lumen structures here and here of HUVEC, indicating the release of GIGSAP tagged VEGF to show its bioactivity. Injection of the GIGSAP hydrogel containing the GIGSAP tagged VEGF promoted angiogenesis here of a mouse stroke model in vivo. So here are the results of confocal laser scanning micrographic observation at the border of the injured core and penumbra, 7 days after the peptide injection. Injection of the peptide hydrogel increased the number of endothelial cell marker laminin positive cells here at the penumbra. Yeah. And we also examined the number of proliferated cells at the penumbra after the peptide injection by EDU assay. An injection of GIGSAP and VEGF GIGSAP significantly increased the number of the EDU here and the laminin positive cells, indicating enhanced angiogenesis at the penumbra. And to evaluate the neuroprotection, we performed FJC assay, staining the degenerating neurons, and NU-N here marker, staining the normal or healthy neurons. An injection of a mixture of VEGF GIGSAP and jigsaw peptide increased the number of the new N labeled cells here at the penumbra, and significantly reduced the number of FJC positive cells, suggesting that the injection of our material suppressed neuron death at the penumbra. And therefore, these histological analysis suggested that a single injection of jigsaw peptide and jigsaw tagged VGF, the mixture promoted angiogenesis and neuroprotection. Finally, a footfall test revealed the improvement of the behavior. So the left-hand side movie shows the behavior of a mouse stroke model that often shows footfold, the left back foot often folds. And in the right-hand side movie, you can see the behavior of the mouse stroke model, actually the same mouse as the left-hand side one, 7 days after the injection of our material. And notably, the frequency of the footfall steps significantly decreased after the injection. And the statistical analysis of the footfold steps indicated that when we injected the mixture of the jigsaw peptide and the jigsaw tagged VGF, so only in that case we could see the significant decrease of the footfold frequency, which is a good result. Yeah, that shows the enhanced recovery of the behavior. So as a summary, in this study, we have developed supramolecular peptide nanofibers that can incorporate the full-length VEGF protein and also release the protein over the 100 hours, which matches the recovery of the injured tissue. And by this function, the supramolecular material can promote angiogenesis and also recovery of the behavioral function of the mouse stroke model. So this approach, we hope that can provide the first and practical cell-free therapeutic modality to treat subacute cerebral infarction. So as an acknowledgement, so most of these experiments in this study were performed Mr. Atsuya Yaguchi, who is one of the PhD candidates in my laboratory. And I appreciate all these collaborators and also funding to support our research. So thank you very much for your kind attention.
Thank you very much, Murakoa-san. Great stuff. Please post your questions, or you can raise your hand if you want to ask the question directly to the speaker by joining the panel. So I have a question. So you have in your jigsaw peptide, you have highly positively charged sequence. You have, I think, more arginine than aspartic acid. So overall, a net charge will be positive. So how does it assemble, self-assemble when you have positive charge? I would assume that there's a repulsive force maybe operating when you form bigger bundle of aggregates.
Yeah, thank you very much for the good question. Let me explain the self-assembly chemistry of our peptide. So the main driving force is the I'm sorry, that there are two main interactions that drives the self-assembly. The first is hydrophobic interaction between the it's an intermolecular hydrophobic interactions between the hydrophobic domain of this peptide. Because this peptide is made of a repeating structure between hydrophilic arginine or aspartic acid residue and hydrophobic amino acid residues. So it shows hydrophilic phase as well as hydrophobic phase across the main chain. And this hydrophobic phase can stack with each other by hydrophobic interaction, and it is controlled. It is controlled by this kind of dovetail packing, like this.
Mhm.
And the second factor is hydrogen bonding between the main chain amide groups, which actually allows for the one-dimensional stacking of the dimer of the peptides. So that in the final structure, the surface is covered mostly by the hydrophilic amino acid residues.
So it's a bunch of dimers.
Yes. So this is actually the kind of the monomer, which stacks into the one-dimensional direction by beta sheet.
So you said that they also form hydrogels in serum.
Right.
I would assume that this charged peptide must be interacting with the lipids in serum. Any
Right. Yeah.
lipids? Do they?
It's quite difficult to study, such a specific interaction in the serum or the physiological condition. Yeah, it might be possible.
Is it a desired effect that you want them to interact with the lipids?
It depends. Quite depends. Yeah, probably it's a good property to make the hydrogel remain at the injected point for a while. But still, for the the precise answering to your question, it's still need more study probably.
I see a question in the Q&A from Beena Krishnan. Do we know how these peptides? Do we know how these peptide or peptide network interact with the natural extracellular matrix associated with cells, especially when injected in the mouse?
In our case, we injected this material into the injured, the injured brain tissue, so that at that region, at least at the beginning, it's like kind of empty. It's injured. So we have not studied such interaction, between the material and the ECM.
What factors control the release of the protein incorporated in these fibers? You said it's about 100 hours. VEGF can be released after 100 hours. And in your in vivo studies, you said it's about 7 days, isn't it? So is there a correlation between these two, or?
Yeah. So the time scale in the in vitro assay and in vivo assay cannot be compared directly because of the quite different conditions. And actually, it is very, very difficult to monitor the diffusion in the in vivo condition. So at least, we can imagine that the releasing time scale should be about 100 hours or a few days, but it's a kind of it's not measured speed. Yeah. And the one of the important factors to regulate the diffusion releasing speed should be the hydrophilicity or hydrophobicity of the material. So as I presented in the previous study by Coulier's group here.
Mhm.
Yep. So they reported the GFP retains inside the hydrogel for a very long time.
Yep.
Yeah. But in our case, we could observe the very slow releasing property, and I think the dynamic difference should be due to the chemical property, probably the hydrophobicity. So the radar 16 has a critical aggregation concentration about 0.023 weight%. And compared to that, our material shows a higher CAC value, which means our material is more hydrophilic. And that property should be one of the points allowing for releasing of the protein, probably inhibiting the non-specific interaction.
Very interesting. So there are two questions in the Q&A folder. You can take a look at it. The first one is from Beena Krishnan. What is the half-life of the injected assembled peptides? How well are these cleared from the system?
Half-life.
Yeah.
You mean gel to Zol transition, kind of that?
Now, how long they can survive in the after injection? What is their life?
In vivo.
In vivo.
So because for monitoring the localization of the material, we have to we have to the surgery of the mouse. So it is technically impossible to monitor the material diffusion using one mouse. So it's a technical limit. However, we monitored the diffusion of the proteins originally incorporated in the hydrogel using the different mouse after injection. So the left-hand side showed the results of the releasing of GFP incorporated in the hydrogel. And in the beginning, after the short time of the injection, we could observe the green fluorescence at the injected point. However, after 72 hours, it was weaker than the observable level. So meaning the diffusion of the GFP. However, in the case of the GFP incorporated in the radar 16, more sticky peptide, then even after 3 days, the GFP, the green fluorescence, was still close to the injected point. So actually the diffusion was suppressed in the right hand side case. So it's it's very difficult to monitor the half-life, but clearly those two materials show different properties. I'm sorry.
But in the in vitro condition, can you not measure the stability against enzymatic degradation?
I see. We have not studied it yet.
Maybe it's quite stable because it forms a dimer and then forms a large bundle of dimers, so it may be quite stable. Then how do you clear these peptides from the system? That's the second question from Bina Krishan.
Well, it's the peptide made of a natural amino acid. So I think the proteases can hydrolyze the peptide, like, yeah. So, and also everything, the assembly is made of non-covalent interaction, the supramolecular. So it does not, stay forever. It collapses over time. So the enzymatic cleavage as well as the diffusion of the supramolecular stacking would be would results in the diffusion of the material in our body.
Just related to that question, so do you have any data on the toxicity of this peptide? Is it toxic at all?
Thank you very much for that question. Yeah. So this is foreign body responses of the GIGSAP as well as the conventional RADA16 material after brain injection. Yeah, we could see the inflammation response in the case of GIGSAP, but the level was quite comparable to the RADA16. And importantly, the RADA16 is a already clinically applied material. So we believe this level should be acceptable for applications.
But the next question is from Ada Caponito. Methionine can oxidize during the several hours of incubation in serum. Does the methionine oxidized peptide self-assemble in the same manner as the original peptide? And does it have the same property?
Yeah, that's a very interesting question. Actually, we are now submitting one paper regarding that point, methionine oxidation. And I hope you are not a reviewer. And let me show you one. Can you see the slide?
Yep.
It's a vertical slide. I'm sorry, so it's quite difficult.
We're seeing it, yeah.
Yeah, so we used chemical oxidation using hydrogen peroxide. So we first made a hydrogel of GIGSAP and added the H2O2 solution on top of that and monitored the dissolution of the hydrogel. Actually, the methionine oxidation is very critical to break the self-assembly of this peptide. So we could observe the gel to Zol, transition over 19 minutes in this case. So, and we in this paper, in this submitting paper, we report that the difference in the gel Zol transition speed depending on the sequence, amino acid sequence. So in the case of the methionine located at the center, at the middle of the peptide, the transition goes slowly. However, when we put the methionine at the terminal side, N or C, in both cases, the gel-Zol transition goes faster, even within 20 minutes compared to 90 minutes. So that is the highlight of this study. But yeah, so in your question, the methionine oxidation is very critical.
Next question is from Velayudham. He says, great talk. Can we use GIGSAP peptide as a mRNA carrier?
I'm sorry, MR?
Messenger RNA carrier. Can you use it for that?
Yeah, might be possible. Yeah, it's a good idea. I would like to try it because the peptide has a positive net charge, and so I believe it can interact with nucleic acid. So as a mixture, we can deliver the composite material to the desired position. So hopefully, and it may also, release the nucleic acid. So yeah, it sounds very interesting.
Are there any other questions from the panel, Danilo?
Oh, yes, just curiosity. I was just thinking about the properties of copper ions as pro-angiogenic factors. So my question is it possible to design histidine-rich building blocks that can help the properties of the VEGF to help to proliferate for endothelial cells? What do you think about this possibility?
You mean the
Building blocks that have copper binding properties, like with histidines, and could be useful in helping the activity of VEGF on the surface?
I'm not sure because the histidine copper complexation would be very strong, which may inhibit the releasing of the VEGF from the material. It depends on the interaction stability or the complexation stability between the histidine and copper. Is it okay? So in our
just an idea.
Many people use actually histidine.
Corroborate the activity of the VEGF. I was wondering if it was possible to envisage something like that.
Right. But yeah,
thank you.
I wonder the copper may be a toxic source. So in our case, we did not want to use copper for
in a free state is toxic. You are true. In a free state, but when it is complexed, it is not toxic.
I see. Yeah, it's a good idea to make a complex between the protein and the peptide. So it depends on the stability.
Yeah.
We have a question in Q&A from Velayudham. What about the reaction between GIG-SAP peptide and H2O2 hydrogen peroxide in the presence of iron 2+? I would say, especially in when you go oxidized.
We have to check it. Yeah, we have to check it. I'm sorry, I cannot answer now.
Okay.